psychiatry. So we now are going to change fields and stop talking about inflammation and immunometabolism for a little while to listen to brain imaging. And I'm very happy to introduce Dr. Charles Laïdi, who is assistant professor in my department in the east of Paris. And his field of research is brain imaging and also brain stimulation. And I've asked him to summarize the literature showing that we could also use precision medicine to stratify patients and identify those that may better respond to brain stimulation. So thank you very much, Charles.
Can you hear me? Yes. So I'm going to talk about how we can use neuroimaging to do stratification and personalization for brain stimulation. So there are different ways of investigating the brain structure. In psychiatry, you can do structural MRI, or you can study the cortical thickness. You can do tractography to measure and to reconstruct the fiber bundles between different parts of the brain. And you can also do functional MRI, which I'll talk a little bit more about today, where you can extract a signal that reflects sort of the consumption of oxygen of the tissue, and then you can compute functional connectivity between different brain regions. So we don't have yet any neuroimaging biomarkers in psychiatry, sadly. What we're seeing is case control differences in large-scale studies. So those are the results from the Enigma studies. As you can see, for instance, in schizophrenia, bipolar disorders, you're seeing a pattern of cortical thinning, but it's not specific, and the effect size is certainly not sufficient to have a robust biomarker. And overall, there are weak brain behavior associations in psychiatry, which is certainly a problem, I think, to develop and advance precision medicine in this field. So I'm interested mostly in non-invasive brain stimulation. So those are methods that, unlike medication where you're targeting the whole brain, you can really try to apply an electric field in a specific region of the brain to target a specific circuit. So I'm going to talk today about TMS, which stands for transcranial magnetic stimulation, and TDCS, which is a current going from one electrode to another that induces a low-level electric field. And the idea is to combine neuroimaging with non-invasive brain stimulation to be able to identify in patients circuits that are disrupted in relation to symptoms, and then to try to target those circuits with non-invasive brain stimulation. So I'm going to talk about different methods to implement precision medicine in psychiatry. The first one being stratification, where you're trying to select the patients that are the most likely to respond to a given treatment. I'm going to talk also about individualized targeting. And this is a different approach because you're trying to target for each patient specifically one region of the brain that might be work well for a given patient. So you're not trying to select the patient, but you're selecting the regions that you're trying to treat in every patient. And then I'll discuss the implications for clinical trials. So I'm going to start with a stratification, which is basically the idea that has been applied in many other domains in psychiatry, that instead of proposing a clinical trial to all the patients, you can define biotypes, for instance, using neuroimaging, but there are other ways to define those biotypes in order to be able to find the right treatment for the right patient. And this approach has been applied to neuroimaging. So this is a Drysdale paper from 2017, where the authors basically studied the functional connectivity in depression, and they did clustering in all those patients with depression to be able to identify biotypes. And they did find an optimal solution of 4 different clusters. As you can see here, there are a blue, a red, and so on. So different types of clusters that are entirely biologically based on the functional connectivity patterns of the brain. Then the next step was to see if those biotypes also were associated with different types of symptoms. And this is what the author were able to show. So you're seeing here anhedonia, psychomotor retardation, and the loading in each of those clusters. So you're seeing different types of depression based on the different types of clusters. And then the authors retrospectively looked at patients that had a TMS treatment across all those 4 different clusters. And they were able to identify that patients belonging to the first biotype were responding to TMS and depression with a rate of 80%, whereas other patients, for instance, from the second biotype, had a response rate to TMS of only 20%. So this paper was very influential and really interesting and raised a lot of hope in the community. But it was based on stratifications on clustering. And the question is it possible to replicate this clustering in other cohorts? The replication is really a huge challenge, I think, in general for neuroimaging in psychiatry, because there are many types of different clustering algorithms. If it's working in a population, it might not work in another population. And a researcher from the Netherlands, I think Brenda Penix was also associated to this work, tried to replicate those clusters in a cohort. And they got an award, a replication award for this work. And they finally failed to replicate those clusters that were originally found by Drysdale and colleague. So I think still stratification is really interesting, but it needs to be replicated in different cohorts to be really robust and stable and to have a clinical interest. So another way of seeing things is instead of trying to select patient, you can select the region that you want to treat in every patient. So here, for instance, you're seeing a functional connectivity map with 2 different targets. So one, the green one, is a personalized target based on the connectivity. So here we're trying to find a region that is hyperconnected in the prefrontal cortex. I'm going to talk about this a little bit more. And the reason why I think that we need those individualized approach in psychiatry is that the response and the remission rates of TMS to treat depression is not that high. So we're seeing a remission rate. We published this paper in the cohorts of 400 patients, and we had a remission rate of 22%, which is not that bad, given that the patient did not respond to a first and then a second antidepressant. But for a treatment, for physical treatment, it's still not very high. So researchers tried to identify ways of individualizing the TMS targets, and they looked at a region called the subgenual anterior cingulate cortex that you can see here in blue that has been associated to depression in many different studies. It's also a target for deep brain stimulation. This is work that has been done by Helen Mayberg. And it's also connected to the DLPFC, which is a region that can be targeted with non-invasive brain stimulation. Because the problem with SGACC is that it's very deep in the brain, so it's not possible to target this region with non-invasive brain stimulation. So instead, we're trying to target the DLPFC that is connected to the SGACC. And several researchers have analyzed, including Michael Fox in the United States, retrospectively some data, and eventually found that when you're targeting a blue region here that is hypoconnected in the DLPFC to the SGACC, you're getting a better TMS outcome than when you're trying to target a region that is hyperconnected to the SGACC. And there've been attempts to try to replicate those findings. I think this finding has been replicated several times. So basically the idea is to calculate what could have been an optimal site of stimulation and calculate the distance between the optimal and the actual site of stimulation. And then you can see if the distance is correlated to the improvement of depression with a TMS treatment. And this has been found by Cash in 2020. 1 year later, it has been replicated by another researcher. And it has been also studied more recently in the American Journal of Psychiatry, where the results were quite a little bit different, but I think the idea remains the same in a much larger sample in 300 subjects. And then Nolan Williams, which is a psychiatrist working at Stanford, had an idea to implement this precision medicine approach in a clinical trial. So instead of stimulating every patient in the same region, which is here represented by the blue dots, he personalized the target for every patient. So here, every dot corresponds to one patient that was stimulated in different parts. He also included an intensive TMS protocol. So instead of proposing 2 sessions per day, he proposed 10 sessions per day and had, quite frankly, outstanding results. So he published first an open study and then a randomized study. And so as you can see here, there's a huge decrease in depression in patients that were in the active versus the placebo arm. Actually, the sample size is quite small, but the clinical trial was stopped halfway because the active arm was so much more efficient than the placebo arm. They decided to discontinue the clinical trial. And so you're seeing here that in the active arm, you have approximately 80% of the patients that were in remission after treatment and only 10% in the placebo arm. So there's really a huge difference, and means that when we're having a huge effect, maybe we don't need a very large sample size to prove this. And we've been able also in the department of Marion Leboyer to replicate those findings. So we're really seeing a clear efficacy of this TMS treatment, intensive TMS treatment in patients with resistant depression. And so this has led to what I think is, to the best of my knowledge, the only precision medicine approach that is FDA approved. So the patients are first getting an MRI brain scan, then they're getting an individualized TMS target, and then afterwards they're getting the actual TMS treatment. The only problem is that we don't know if the efficacy of this protocol is related to the 10 sessions per day or to the individualization of the target using MRI. You can also use neuroimaging in other ways to study non-invasive brain stimulation. So you can do what is, I think, the dream of a psychiatrist. So when you're prescribing a medication, you would love to simulate what would be the effect of the medication on the brain of your patient. So this is possible with simulation softwares, where you can basically simulate the electric field and how the electric field is applying to the brain in a given individual. And so we've been working on doing a meta-analysis combined with those simulation studies to simulate the electric field in every study and see which brain region would be associated with a better outcome. So as you can see, if you're using different types of TDCS montage here, so you're seeing the 2 electrodes, the electric field is different. But what is also very important is that there is a large inter-individual variability. So here are 8 different subjects, and you're seeing that the electric field is very different in the subject, although the electrodes were placed in the same regions. So maybe this opens to the idea to, instead of treating all patients in the same way, to have a first phase of simulation in the patients where you can adjust the electrodes and adjust the electric current to be sure that you're really engaging the regions that you're interested in. And in this work, we were able to show that some brain region in the prefrontal cortex were associated to a better TDCS outcome, but those were not the regions that were stimulated in every patient. So I think that there's really a room for improvements by using brain stimulation in this domain. And so finally, I'm going to talk a little bit more about ongoing and clinical trials that have been published. So very recently, there was a very large clinical trial published in Nature Medicine comparing more than 100 patients that received a connectivity-guided TMS treatment and that were compared to the same amount of treatment-resistant patients with a regular approach. So this was published this year in Nature Medicine. And we were all very disappointed to see that there was no significant differences in both arms. So the question is this a field killer, as Andy was mentioning yesterday? I'm not sure, but I think that before starting clinical trials, implementing precision medicine approach, there might be some step that we need to follow, or some level of evidence that we need before getting at that stage. And I think the first question is to know whether there is a basis for personalization. We need to have a signal that is related to the clinical outcome, but also a certain level of inter-individual spatial variation, because otherwise it doesn't make any sense really to personalize the treatment. We need also to have a reproducible way of doing this kind of targeting, to be sure that if you're scanning a patient several times, you're getting the exact same target every time that you're scanning the patients. And so we've been working on this to implement an open toolbox to implement this kind of precision TMS targeting, because the problem is that a lot of research are using, custom codes that are not public, so it's really hard to compare the different types of codes. So we're working in an open code to be able to implement this precision targeting. So we're using consensus method. We're also injecting noise to see how the clustering is going to be robust to noise. So hopefully we'll be able to release this toolbox soon. And you need also to be sure that when you're doing the precision targeting, there is enough inter-individual heterogeneity. So you're seeing from here that in the CASH paper, the targets are well distributed in the DLPFC, but in the Cole paper here, you can see that all the targets are more or less in the same area. So we need to be sure that there is a large diversity, because if we're stimulating in the same brain region all the patients, obviously it's not a precision medicine approach. And last, I think also we need sufficient amount of retrospective data analysis showing that there is room for personalization. And so I think that this has been the case. So Cash, Siddiqui, Erbao, and D'Souza recently in a preprint, they all showed that the connectivity pattern of the TMS target matters for the TMS outcome. So I think those are really strong results. And hopefully this will open the way for an RCT with prospective personalized targeting, and maybe also in the framework of the PEPR PROPSY. So thank you very much for your attention.
Thank you very much, Charles, for an extensive review, good questions, and I think ongoing clinical trials that are going to answer some of them. So thank you very much.